So I guess people are in the room now. So a very warm welcome to the second day of our workshop on spatial transcriptomics data analysis in Python. Today we have 2 sessions and a closing talk by Johanna Klughammer. And the first session will be by Alma Andersson, and she will present the tool Eggplant. So again, similar as yesterday, a first and theoretical introduction into the tool, and then followed by a live demo on the tutorial. We will use the same Slido as yesterday for raising and voting on questions. I will post the link in a minute, and then again, you're very welcome to raise questions on Zulip or use the GitHub repo for questions on install issues. Yes, so I would hand over to Alma and stop my screen sharing.
Thank you, Anna. So I'm just gonna get started with sharing my screen here. Let's see. I'm just gonna do some configurations here. So can you see my slides?
Yes. Yes.
Awesome. Yeah. So thank you for the introduction, and I'm super stoked to be kicking off this second day of the workshop, which I think is a really nice initiative. And as said, I'm going to be presenting on this tool called Eggplant and this idea of building common coordinate frameworks for spatial transcriptomics data. Before we get started with that, though, I just wanted to give you an outline of the agenda or my idea for this session that I'm supposed to host here. And I wanted to start with just introducing the problem, trying to explain why we need a method like this and what's relevant about it, and the problem that we're trying to address, to then present a solution to this problem and outlining how our approach works, that being Eggplant, to then show you some results, giving you 3 examples that highlights different aspects of Eggplant and the type of analysis that you can make with it. And then I think that will round off the theoretical part, and I think that's a good time to have some questions addressing things related to the presentation. And once that's done, we can continue on to do this walkthrough of the notebook, getting familiar with Eggplant. And of course, then you're free to ask questions on Tulip or use via Zoom, I guess. So yeah, with that being said, let us set the scene a bit here. And you can imagine a fairly common scenario that we often encounter in our projects or experiments where we have collected tissue samples representing the very same structure, but from multiple different sources, such as, for example, different conditions, case and control, or different time points, for example, along the developmental trajectory, or maybe just have different replicates from different individuals to get more statistical power. And very often in these projects or experiments, one of our main objectives is to compare and relate features between our different samples. That's the whole point of gathering these samples. And I guess some examples of projects where this is extremely relevant are these atlas efforts, for example, such as the Human Cell Atlas. And in order to achieve this objective, when we're working with non-spatial data, we tend to employ these strategies for batch correction or data integration. And we then do this by creating this low-dimensional embedding based on the gene expression, and then try to relate observations in this low-dimensional gene expression space. However, when we're working with spatial data, what's super cool here is that our data already possesses an inherent low-dimensional representation, that being the spatial domain. And this allows us to extend this idea of integration beyond just the removal of batch effects, but also includes the ability to relate observations in the physical domain. And this is super cool, but it has certain challenges attached to it. And I think the biggest one here is this issue of having independent coordinate systems. And that is an abstract concept that I think is best explained by giving a brief example, which I intend to do here. So you can imagine that we have collected 2 tissue samples from different time points, time point 1 and time point 2. And our objective here is to examine how the gene expression at a certain position, here marked in green, it changes over time. And the obvious issue here is that the coordinates x and y, they don't represent the same position in time point 1 as in time point 2. And that's because we have 2 independent coordinate systems. And obviously, the correct approach here would be to compare the expression at coordinates x and y at time point 1 with the expression at coordinates x prime and y prime at time point 2. And one way of addressing this problem is to transfer the expression to a common coordinate framework where the coordinates x and y actually do represent the same position in all of the different samples. And I just want to take a moment here to elaborate a bit more on this idea of common coordinate frameworks, or CCFs for short. And essentially what we're doing here is that we take some sort of associated information, for example, the gene expression or cell type proportions or protein levels that we have associated with our spatial data. And we transfer this information to a reference, a common coordinate framework that represents the same structure for all of different samples. And I just want to sort of like emphasize here that the transfer data, it's sort of like more than just an alignment. The features are actually cast into the same reference or common coordinate framework here. And it also allows us to correct for nonlinear distortions, not only these linear distortions such as translation or rotation, for example. And it is sort of like this transfer process here that we've sort of like been focusing on trying to develop a method for. But before I get into the details of the specific method, you might sort of like think that this doesn't seem like super exciting. Like, why would we have the data sort of like look like this where everything's used the same? And that's kind of true. But you can also imagine a slightly more interesting scenario or different sort of like condition here where we have some sort of like samples where we expect to see a difference in the spatial gene expression, for example. And casting the data into the same reference, we can compare the expression location by location between our different samples, such as in this case here, where we can see how the gene expression pattern differs quite a lot between individual 1 and individual 2. And this allows us to assess the differences both visually, but also computationally. And I guess an example of what sort of like scenario this could represent is for example, if individual 1 and individual 2 represents different ages, or maybe it's sort of like case versus control. So I should also sort of like mention here that very often just creating these CCFs or references and transferring data to it is not really the end goal of our analysis, but it rather enables plenty of really cool and interesting downstream analyses. And just to give a few examples here, we can sort of like do this spatial temporal modeling, for example, once we cast the data into the same reference, where we analyze and characterize how the expression changes over time and space simultaneously. We can also look at local expression changes, where we assess whether expression is up or down regulated in certain regions. And we can also do this sort of like aggregation of data, where we jointly represent information from multiple samples by a composite or average representation. And this in turn allows us to identify general spatial expression patterns found in the data. And obviously, there's a lot more things that you can do once you sort of like made this transfer to the reference, but I think these are 3 fairly relevant and interesting examples that are worth to highlight a bit. I've been speaking a lot about sort of like the problem here and why we need a solution. And so what is the solution here? And maybe I should sort of like change this statement here and say, what is our solution? Because as always, there are multiple different approaches to tackle every single problem. But our approach is sort of like this Eggplant tool here. And it stands for effortless generic GP landmark transfer. And there's sort of like a nice story behind this logo here that I usually tell, that it used to look very different before. It was very, very heavily inspired by the eggplant emoji. But then I had a pretty sort of like big talk, and one of the very kind audience members were like, maybe you would like to consider changing this logo because it could have certain unwanted associations. So this is like the second iteration of the logo, and hopefully now it should be sort of like free from having any such unwanted associations. So let us have a look at the method itself then. And you can basically think of this as a 5-step process, where you start off by constructing or choosing a reference that you want to transfer information to, and then you select samples that you want to transfer information from to this reference. Next, we chart the landmarks in our data, and that is basically annotating common landmarks found both in the observed data and in the reference. We then select a feature of interest, for example, the expression of favorite gene that we want to transfer from the observed data to the reference. We then learn a transfer function relating landmark distances to the gene expression. And finally, we transfer the information to the reference by applying this learned transfer function to each location within the reference. And now there are 2 parts here that I would just like to spend a bit extra time on, because maybe it's not super obvious what I mean with them, just from this very brief introduction. And those are the landmarks and the transfer function. So let us start off by looking at sort of like the landmarks here. And yeah, so maybe the most fair question here is sort of like, what is even a landmark? And landmarks are sort of like structures, signals, or any features that are easily identified across the different samples. And this definition is largely based on sort of like what is presented in this really nice publication called Toward a Common Coordinate Framework for the Human Body by Rood et al. And if you're interested in this concept of a common coordinate framework, I can really recommend this publication. And what's sort of like key here is that a landmark should always represent the same approximate position within the different tissue samples or samples. And the landmarks can be basically based on anything that helps you to sort of like identify them. And for example, if you're working with ST and Visium data, you might have an image representing the morphology of your tissue that you can use to guide your annotation. But you could also use the gene expression itself. Perhaps it's like by visualizing certain genes, you would be able to pinpoint certain locations within your tissue, or maybe have some sort of like orthogonal protein signal staining, for example. And what is kind of important here is that Eggplant only focuses on the transfer process. So it doesn't really do anything to contribute with annotation. So you can call it as or think of it as an annotation agnostic method, basically. And all of the results that you will be seeing very soon here, they have gone through sort of like a manual landmark annotation, and that's sort of like done by a non-expert, also known as me. And that's actually quite encouraging, because if I manage to annotate these samples and get the method to work, anyone with a slight sort of like knowledge of the biology should be able to do it even better. But we're also working on an automatic approach that does this landmark annotation automatically for you. And now I see here that the graphics are a bit messed up because I was supposed to give an example of a good landmark here, and the circle was supposed to point to this tip of the dentate gyrus. So you can sort of like ignore the circle, but look at the arrow instead. And basically this would constitute a good landmark because it's sort of like an easy point to identify in all of the different samples. And assuming that we're at the same depth within the brain, we can sort of like think of this as representing the same approximate position in all of our samples. So those are the landmarks. Let us then move on to the transfer function here. And this is sort of like the core part of the actual tool here. So essentially what we do here is that we first look at observed data, and for each location, we measure the distance to all of the different landmarks, creating this distance matrix X. We then assume that the gene expression Y is related to the landmark distances via transfer function F. And we also assume that the transfer function follows a Gaussian process distribution. And this allows us to use Gaussian process regression to determine the character of the transfer function. Next, we move on to the reference, and we do something very similar. We again create this distance matrix X bar, but this time for the reference, where we measure the distance from each location within the reference to the different landmarks within the reference. And we then transfer the information from the observed data to the reference by applying this transfer function that we learned from the observed data to this X bar here. And this allows us to predict the gene expression at each location within the reference. And here you can sort of like you see a flow of the information and just get an overview of the model. So I've been speaking quite a lot about the theoretical part and I just want to make some brief statements here about the implementation. So this is sort of like implemented as a Python package, and it's written to be compatible with undata objects, and that's sort of like how we expect you to sort of like have formatted your data. It has a very scanpy or squidpy like APIs, which if you're familiar with any of those tools, it should be fairly straightforward to use Eggplant as well, as you will see later on. And it can be installed using PyPy and GitHub, and I would perhaps recommend it in sort of like in the future to use the GitHub installation because that's slightly more updated usually, and it's also documented on read-the-docs. And as a backend, we're using sort of like PyTorch or gPyTorch, which is really helpful because it saves us a lot of time from implementing these Gaussian processes ourselves, and it's really efficient. And speaking about Gaussian processes, if you have any experience with them, you know that they have sort of like this notoriously bad scaling. So the complexity is O(n)(3), where n is the number of observations. And we have plenty of observations in our spatial data. For example, in slide-seq data, we can have up to like 50,000 different observations. And we also have quite a lot of different features. So that, of course, introduces a challenge to sort of like making it scalable to large data sets or to even our data sets, being sort of like this large transcriptome-wide data sets. And we have developed certain strategies to sort of like tackle this challenge. And I'm briefly going to go through 3 of those strategies here. And the first one is fairly straightforward. It's usually just like use GPU acceleration, which gives a general acceleration of the inference process. And that's completely handled by PyTorch and gPyTorch. So I'm very thankful for that sort of like framework. We're also using sort of like variational inference with inducing points. And this sort of like reduces the complexity with respect to the number of observations from O(n)(3) to O(m)² times n, where m is the number of inducing points. And here I should give a really big shout out to Giovanni, who actually sort of like was the one who made me implement this. It was on my to-do list, and he was like, is this something that you could potentially implement? And I was like, yeah, let's take it off the to-do list and actually do it. So yeah, he's sort of like the one who catalyzed the process here. And we also have this sort of like fast approximate transfer alternative here, which uses a data decomposition step. And it reduces the number of features that has to be transferred when you want to work with, for example, the whole transcriptome and sort of like want to transfer every single gene from the observed data to the reference. And it's essentially a 3-step process here where we use PCA to project the full dimensional data into a k-dimensional subspace. We transfer the loadings of each principal component to the reference, and then we sort of like reconstruct the full dimensional data within the reference. And this, of course, comes at some cost of accuracy because we get this reconstruction loss, but it's really efficient. It sort of like reduces the time that we need to sort of like spend on transferring every single gene quite significantly. So that's one alternative as well. So just to speak a bit about the input and output here. So essentially you need your undata objects containing the feature of interest that you would like to transfer to the reference. You also need the coordinates for your different observations, for example, the spots here. And you also need to have the positions on the different landmarks. And actually those positions aren't explicitly used in the transfer of the information, but we rather use these distances, but we need the positions in order to compute the distances. And you also then need a reference object, which is sort of like built on this reference image that you define to sort of like represent the reference itself. And we also have the reference landmark coordinates telling us where in the reference we have the different coordinates. And this uses input to Eggplant, and it will sort of like spit out this sort of like updated reference object, which also contains an undata object where you have sort of like the different features that you transfer to reference all represented within the said reference sort of. So that's the input and the output. So I just also want to show you some brief examples here of the analysis And for that, I've sort of like chosen 3 different data sets here, one of the human developmental heart, one of the mouse brain in the sagittal section, and one sort of like of the perturbation in the mouse brain. And these are sort of like chosen to represent different aspects of the eggplant analysis that I think is sort of like nice to perhaps highlight and put some emphasis on. And also sort of like included here a comparison with sort of like an alignment-based method, because sometimes it can be a bit confusing what's the difference with these sort of like transfer-based methods and when we have this alignment here. So let us get started with the human developmental heart. And here we have 7 different sections. And as you probably can see, they stem from 2 different individuals, individual 1 and individual 2. And below here, I can also actually say here that we have marked the landmarks here with these differently colored stars. So that's sort of like the marker that I've chosen to use to indicate landmark positions in all of the different samples. And below here, we have sort of like the reference that we want to transfer the information to. And you can also see how we annotated different regions here in the reference. And for now, you can sort of like actually not think too much about them, but I will come back to them later on. And in this case, the kind of information that we want to transfer is the expression of 4 different genes, MYH6, ELN, MYH7, and COL2A1. And initially, when I was looking at this data set, I was a bit concerned as to whether it would actually work, because if you look at individual 1 and individual 2, you see some quite distinct differences in the spatial structure between individual 1 and individual 2. But Eggplant is a very easy method to apply to these data sets, so I did it just like to see what happened. And actually, the results were quite encouraging. We could see some good agreement between samples from the different individuals, so that was really positive. Still, this is quite a lot of information to like take in and digest. But what's really nice is now that we have sort of like all of the information from these different samples cast into the same reference, we can create these composite representations that allows us to summarize the information across all of the different samples. What we also did then, sort of like as I guess quality control, was to look at the location-wise correlation across the 7 different samples. And here we saw something really positive. We saw that we, as expected, had a high correlation between sections from the same individual, but also between sections from the different individuals. And that's really nice as well to see that, meaning that it seems like we actually managed to transfer the information from the different individuals in a really good way. And now coming back or like revisiting this reference here, where we have sort of like annotated different regions. So we didn't annotate anything in the observed data, only in the reference, but this allows us then to actually sort of like do a form of spatial enrichment analysis where we can assess whether certain genes are up or down regulated in specific regions. And one thing that we noted here then was, for example, this left-right asymmetry in the COL2A1 gene, which is sort of like in the left upper corner there, where there seems to be a higher expression of this gene in one of the atria compared to the other. So there's like a higher expression in the blue atrium compared to the yellow one. And this sort of like analysis is quite useful sometimes as well, at least in my opinion. So to finally just like make this sort of like comparison between an alignment-based method and a CCF-based method, what I did here was that I took 2 sections from each of the different individuals. So section A and section E here, and then we used sort of like this method that was recently published in Nature Methods called PASTE, which is super nice and it allows you to align your spatial transcriptomics data based on both sort of like the spatial structure of your data, but also the gene expression. And that's exactly what we did here. We aligned these 2 sections. And you see that the alignment works pretty well, but what's kind of important to emphasize here is that the coordinates, they still don't really correspond to the same position. And you can, for example, see here how a part of the atria in the red section sort of like overlaps with the main body in the blue section. And that's because we're only aligning these existing sections. We don't really cast the data into the same reference. While if we're using sort of like a CCF-based method, we actually do get sort of like the data put in the same space, and we get these corresponding coordinates that I tried to sort of like mention before. So that's about the developmental heart. Now, for the mouse brain here, the sagittal section, this will be a very brief sort of like example here, but what I really want to sort of like highlight here is how sort of like with this transfer process, we actually managed to preserve these very complex structures that we see in the gene expression patterns. So here we have 2 sections from the mouse brain. It's one of the sagittal sections, and they look almost identical, but there are some slight differences here, but they are consecutive sections, so you wouldn't expect to see too much difference here. And in the middle part here, you can sort of like see the observed gene expression of 3 different genes in the 2 different sections. And to the right, you can sort of like see the reference that you want to transfer this information to. And now this is what happens when we transfer the gene expression. And as you can see, we sort of like actually managed to preserve these fairly intricate structures. And I think that's quite encouraging as well, because in the developmental heart, we didn't really see that. And that's because we didn't have those sort of like complex structures there to even try to assess whether we managed to preserve them or not. And finally, going on to this sort of like perturbation in the mouse brain analysis, I wanted to show like maybe introduce this concept known as spatial differential expression analysis, or SDA for short. And here we're using already published data from a fairly nice study where what they did was basically inject mice with partially nothing as a control, but also like a saline solution as a sham, and then also like with a heme solution, which was sort of like the active compound that they wanted to investigate the effect on the mice with sort of. They did quite a lot of different stuff here, but they also produced sort of like this Visium data from these mice that I used to mention. And below here in sort of like the image, you can sort of like see an excerpt from one of their main figures, just like very briefly outlining the experimental setup, I guess. And we're not right now, at least, super interested in the biology, even though that's probably really cool. But what we are interested in is it's all like this actual data set where we have sort of like different conditions or perturbations from the normal state. And we have sort of like, as I said, the control, we have the heme case and the sham case. And here we have the expression of 3 different genes that they mentioned in the publication. And again, we have a reference to the right here that we want to transfer this information to. And just as we did before, we can make this sort of like transfer of information, and it seems to have been working pretty fine. The data is now also like represented in the very same reference, and maybe it becomes a bit more clear here now than in the previous example where the 2 sections were very similar, but here we can see that there are some spatial differences in the different cases, for example. So I now want to illustrate this SDA, or spatial differential expression analysis, using this gene here, SERPINA3N, and I just took that one because it looked pretty nice. And basically what we're doing here is that we compare the spatial gene expression across the different conditions. So for example, if you look at this middle part here, where you see that there's a red signal, we have sort of like a higher expression of this specific gene, SERPINA3N, in the heme case, while there's sort of like a blue signal, we have a higher expression in the sham case compared to the heme case, and vice versa for the different combinations of cases to the left and right here. And exactly how we're doing this is very straightforward, actually. We just sort of like compute the difference between the 2 different conditions using something that I sort of like call spatial arithmetics, and this is just like subtracting the expression point by point. And this gives us this sort of like location by location comparison. And I can also say here that the gray regions, they represent non-differentially expressed expression levels, or like differences between these 2 different cases. And maybe something that's worthwhile highlighting here is sort of like that this is only possible when we have the data inhabiting the same reference or common coordinate framework. So to summarize this theoretical part a bit, a common coordinate framework, or CCF, represents sort of like a unified framework that allows us to relate observations across different samples. And implicitly, this also means that we can make comparisons across time points or conditions. And transferring data to a CCF is not really the same thing as aligning samples. Just want to emphasize that a bit again. And then we have, for the spatial data, spatially aware CCFs, which doesn't only focus on the gene expression, sort of like information can be highly useful. And our method, Eggplant, uses sort of like landmark annotations and Gaussian process regression. Those are sort of like the core elements of our method. And this sort of like final thing that I introduced here, spatial differential expression analysis, is sort of like enabled by having the data in the same reference or CCF, and it represents a way to compare the expression location by location. And if you're interested in to sort of like read more about this method, you can check out the bioarchive preprint. There's also like a publication in review in Nature Methods, so hopefully there will be a publication for you to check out as well, fairly soon at least. There's a GitHub site where you have some information, installation instructions, and there's also sort of like this reader docs page that you can access where we have fairly extensive documentation of the package. And with that being said, I just want to make some final acknowledgements here, saying thank you to Jeanette, Shafay, and Erik, who produced the developmental heart data for me, some code beta testing, and some nice discussions with Paula Tronewski and my supervisor, Joachim Lundeberg, as well as the whole spatial research group that I'm a part of. And again, a bit of special shout out here to Giovanni for just like giving some really excellent feedback and being one of sort of like the first people to actually use this more extensively. So thanks for that. And of course, thank you to all of you for the attention, and I think now is a good time to take some questions. Perhaps I haven't been able to keep up with the Zulip chat or this Slido, but maybe
I can also address the questions. We have a couple of questions in the Slido, so I can just read them out to you.
Yeah, sure.
The first one was, you made me curious. Can you show us the original Eggplant logo, please? So after you mentioned
yeah, it's been discarded. It's deep down in the non sort of like to display folder. Yeah, maybe I can find it somewhere. I can post it in the Zulip's chat then.
Yes. The second one, which was also raised in the Zoom channel, was how many landmarks are needed? How's the correction of nonlinear distortion archived?
Yeah, super good question. So we have sort of like a more or less heuristic approach to estimate the number of landmarks that's implemented actually as a function in the package. But I would say between 5 to 10 landmarks, or like 5 to 20, depending on the sort of like intricate structure of your tissue, not really the gene expressions complex structure, but just like if you have the different holes or maybe different sort of like shapes of the actual spatial domain, you can need like a slight bit more landmarks. But 5 to 20, usually actually 5 to 10 is sufficient. Sometimes I use more because it's easy to annotate, but it's not like a lot of landmarks are actually needed then. So like, of course, the more landmarks you have, the better the performance will be, but it's sort of like these marginal gains in the end. And that's something that you can assess sort of like within your data. We have sort of like functions that support that. What was the second question?
The second one, let me check. It was, how is the correction of nonlinear distortion archived?
Yeah, right. So partially we do that by using these Gaussian processes and actually modeling the expression as a function of the distances to these different landmarks. And also in sort of like in the pre-processing steps, we use some of these thin plate spline transformations just to make sure that we sort of like get the correct measurements between our different landmarks and observations. So those are the two sort of like core elements that actually achieve that.
Okay, nice, thank you. The next one was, could Eggplant handle grossly distorted tissues, like for example, tumors or appearing growths? Is this the appropriate tool for such a task?
Yeah, I think that's an excellent question. So I would say that Eggplant is perhaps more suitable when we're working with sort of like these stereotypical tissues where we expect to have sort of like similar, maybe, you know, structures, but there might be differences in the gene expression. Tumor, to me, sort of like represents a highly perturbed tissue. Like I think one of the hallmarks of tumors is sort of like that there is no structure, and like every tumor is sort of like unique in its own way. So it might be hard to actually try to cast it into the same reference, because what would that reference even represent? So I would say that perhaps just the tumor itself wouldn't really be the optimal sort of like case of using this. However, if you have a stereotypical tissue domain or similar where you have a tumor sort of like perturbing the normal state, maybe that is something that you could compare with sort of like a control state, for example. So it depends a bit on the use case, but sometimes I think cancer can be quite complicated to use with Eggplant.
Yeah. The next question plays into a similar topic, I would say. It was, can you transpose data from two modalities onto a common coordinate framework, say Visium and Murphish, so not the whole transcriptome from different individuals?
Yeah, so let's see if I got this question correctly. The question was maybe like if you can combine different modalities from, for example, tumor data. So since I answered sort of like a tumor aspect, I'm just going to address these sort of like different modality questions. And that's something that you can do is try to sort of like combine slide-seq data and Visium data, for example. Those are slightly more similar, of course, than Visium and Murphish. But if you do the data normalization correctly, so I can also mention that we sort of like assume that the data that you give as input to Eggplant has sort of like been batch corrected for and then everything. So that's not really something that we bother about because other people work quite extensively with that as well. So if you apply these sort of like normalization strategies or integration methods and then try to transfer your Murphish and Visium data to the same CCF, that should work completely fine as well. So theoretically, yes, I haven't really tried exactly that specific combination, but I don't see any sort of like thing that would prevent it from being possible.
Great, thank you. The next question would be, if you evaluate the alignment only at the level of the landmarks, do you assume that all regions in between are automatically aligned correctly?
So that's an interesting question. I'm not sure exactly if I understand. So like if there's a question, if we did that, or if it's sort of like a theoretical question, if we were to do that, because we don't really evaluate just the landmark, so like alignment based on the landmark positions. We look at every single point more or less. So maybe that person could either maybe elaborate a bit more on the question, or just contact me on Zulip and also try to give the best answers possible.
In general, if your question of the participants, if the question wasn't answered fully, then just raise it again and we will address it again.
Yeah.
Yes. And then I think the last, at least in Slido we have is, what if the spatial slice is a cut at a different angle than the CCF space slice? You were showing the CCF as a 2D slice as well. You use as a target.
Yeah, so that's interesting. So that's a quite common question, and I guess it's sort of like the larger question here is sort of like, is this sort of like plausible to work within sort of like 3D models, for example, where you maybe have different sections cut at different angles and want to sort of like translate to the very sort of like same 3D model somehow, or like 3D reference. And so like looking at the core mechanics, there's like no limit actually to which dimension. Like we could work with the 5th dimension as well. It's just like measuring distances, even though that would make no sense obviously to have 5 dimensions of tissue. But right now the implementation only supports 2D reference. references. I think that's something that I'm going to change. It's just like modifying some, I guess, slight pieces of the code. It's sort of like the visualization part that I would have to work a bit more on. But there's nothing so like theoretically that prevents this. It's just that the code is not quite there yet, I guess.
Yes. And then I see we have 2 more questions in the chat, so I'll just raise them again, and then we can maybe continue with the tutorial and raise the remaining questions afterwards.
Yeah.
So the question would I be wrong in saying that paste is more intended for the 3D reconstruction from the same sample, like a serial selections?
Oh yeah, so I really don't want to speak for another developer's method, but if I were to give my interpretation of it, at least I think it's a fairly good interpretation that the person who asked this question made, that it's really good to use when you just want to align very similar sections, perhaps if you have replicates, for example, of your own tissue where there's basically no difference in the morphology. For example, the sagittal mouse brain sections that I showed before, it's a perfect case. And if we assume that there's only slight perturbations in sequentially when we're working in 3D, so if you were to make slices from a 3D piece, that maybe there are some differences, but not that much if you have adjacent sections, that would be one way of aligning your 3D sections. However, I don't think that's really applicable to all tissue types, because, for example, in the brain, we see quite a lot of differences the further in we go. But yeah, I think that's a fairly good interpretation. Yeah, definitely. Yeah.
And maybe the last one before we jump into tutorials. And how do you transfer based on gene expression if there are huge differences between samples caused by pathology? For example, in sample 1, the expression of that gene is very high, while in another sample, it's low or non-expressed.
Yeah, so I mean, that's a good, so let's see here, if I interpret the question correctly. So it's not really about transferring lowly and highly expressed genes, it's about using the gene expression to identify your landmarks or not. Let's see, I can answer this from both angles, maybe, so maybe we get at least maybe one answer correct. So we partially normalize the data, so making this normalization also a standard scaling before we transfer it. But obviously then if you have a more lowly expressed genes, that would actually be something that showed up in, for example, the spatial differential gene expression analysis. So I think that's actually a feature of the different samples that should be different, right? Because that's something that, unless it's a technical artifact, and then the normalizations should account for that. And if it's the question pertains to actually annotating the landmarks using the gene expression, if it's very lowly expressed, that might be hard to rely on the gene expression. I would perhaps instead use the morphology or a protein signal. So it's not that one time, it's not always going to work to use the gene expression, but sometimes perhaps. And maybe you could use a different set of genes. If you have a Visium data set, you have 20,000 more genes to choose from. So maybe one of them shouldn't be that perturbed by the disease state, perhaps.
Yeah, perfect. A note to the participants, the remaining questions we can ask in the live Q&A after the tutorial, just for the sake of time. And I will note all of them and address them later. And I saw additionally, again, install questions. So please raise them in the Zulip or on the GitHub repo. It's much easier to answer them if we have some chunk of code where the code fails or the install process fails. So that would be really, really great. Thank you. Then we can jump now into the tutorial session, and please keep asking questions on Slido, Zulip, or the chat. Thanks.
Yeah, awesome. And thanks for forwarding the questions to me. That's super nice. So let's see here. I think I have one more slide here just to say something about the notebook walkthrough. So basically what we're going to do in this notebook is to just look at how we can load the data and the references, how we prepare the data and the references for the transfer of the information, and also actually making this transfer information to the reference using 2 different strategies. One being this exact strategy that is slightly slower but more accurate approach, and then using this fast approximate approach, which is significantly faster but at the cost of some accuracy. And we're also going to be looking at a way of creating these composite representations and then doing some regional enrichment analysis, and then finally rounding it off with this spatial differential gene expression analysis, or FDA for short. And the data sets we're going to be working with is this human developmental heart data set from our study, and this perturbed mouse brain that I was showing in as the last example of the analysis here. And I just want to make a brief comment, and I'm going to say this again, but just to sort of like highlight that you can run this notebook in something that I've sort of like dubbed demo mode. And this allows you to actually click through all of the different cells, but not really run all of the analyses because it can take some time. It usually takes like 30 seconds to run one single transfer. We're going to do quite a lot of transfers here. So by running it in demo mode, you're just going to do a few epochs, and then we're going to after sort of like afterwards load some pre-analyzed data that already has all of the results contained within it. So with that being said, I can move on to actually opening the notebook here. The first part here, excuse me, if I take some water. The first part here, so like just contains the information that I was going through in the presentation, very sort of like more in a more condensed format. But it was just in case someone didn't listen to the presentation, or if you want to do this later on, I don't remember what I said. And there's a bit of a dictionary and some comments here, but essentially we can just start off by loading some necessary packages here. And hopefully that doesn't take too much time. There we go. I'm going to set this demo mode to true, and I'm also going to set the device here to GPU because I'm fortunate enough to be running this on my GPU server, or not my, but the lab's GPU server. This save object variable here just tells us whether we want to save our result and overwrite the current results that we can load in as being pre-analyzed, and we don't really want to do that right now. And I also don't really want to download the data because that's something that I've already done to save some time. But if you want to run through this notebook, you can simply just put download data to true and then run this cell below here, and hopefully it should download the data for you. If it's not doing that, you can just click on one of the links here and you will sort of like see the data here in a data.zip folder, and you position that in this at the same sort of like in a folder at the same level as the notebook directory, more or less, and it's going to be able to load in all of the data. And I've only tested this on a Linux computer, so I'm not sure if it works that well on Windows computers. Those always seem to be a bit funky. So that's sort of like why I included this link for the manual download as well. So with that being said, we can sort of like just define some of the paths where we're going to load our data from, and then we're going to read in these sort of like undata objects, and we're going to give them, we're going to store the undata objects in a dictionary, and we're going to give them some names that is called like DHX, where X is sort of like replaced by a letter from A to G representing the different sections. So let us load the data and ignore that warning. And here you can sort of like see the dictionary that's containing all of the undata objects, and that's sort of like the data that we're going to be analyzing here first. So we can use now to make sure that everything loaded correctly and such, and that we have the landmarks where we want them in our data. And as said before, we sort of like expect the data to already be annotated. So it's something you have to do yourself. We can visualize these data sets, and we're not really interested in any specific feature or gene right now. So we're going to put features to none. We would like to look at the image since we're working with Visium data here. We can plot the H&E image, and yeah, we're interested in the landmarks as well. So let's visualize this. And that is done using one of the plot functions called visualize observed from the Eggplant's plot module. And here we have the 7 different sections. So that looks nice, and we have the landmarks included there as well. So let us then move on to load the reference, and we're going to load the reference image, and we're going to load the reference landmark coordinates here. And we can also have a look at the, for example, coordinates here, so you get an idea what that looks like. It's basically a list of the different coordinates and its positions, and we have 16 different landmarks here. So just to make sure that the landmarks and the reference looks okay, we can visualize them. And again, it seems like everything's working fine. The landmarks are positioned at the same position as we would expect them to be. So now, once we loaded this reference image, we want to convert it to a grid that we can use to sort of like create this reference object. And for that, we have this sort of like pre-processing function in the pre-processing module that's called reference to grid. So we just like give the reference image here, and give the number of approximate points that we want in our grid. And usually this is sort of like a good recommendation here is to put about the same number of points as you have spots, for example, something in the same order of magnitude. I know that there's like 5,000 spots in the Visium array, but I put 10,000 here because there's going to be some fallout as well. And the number of regions here is quite important, at least in this case, because we have 4 different regions colored in different colors here. So we want to specify how many regions there are, and also tell which color there is of the background has. In this case, we have a white background in this image, and that's just for us to be able to segment it into different regions of the grid. And that's going to run, and we get this DH grid, and we get the DH meta data here. And the metadata just tells us to sort of like which region the different grid points are related to. And now these different regions are just called something from 0, 1, 2, 3. So we are soon going to give them some more informative names, but for now we can just like start off by visualizing this grid that we just created. And again, it looks like we have a good correspondence. The grid creation went okay. Usually the grid creation goes just fine, but I think it's always nice to make sure that nothing unexpected happens when we create our data here. And here we're just going to rename these different regions to something that's a bit more interpretable. So we're going to give this red part here, calling it the main body of the heart. We call number 1 here the green part, the outflow tract, and then we call these 2 different atrium 1 and atrium 2. We're going to run this cell, and we can have a look at the metadata that we just updated now. So I have the region ID and the new names here in the region name. And from that, we can now create this reference object, and we use the grid as input. We use the landmark positions that we loaded before as input, and we use the metadata that we just created together with the grid. We get this reference object here. So we now created a reference and we've loaded the data, which is super nice. So next up is sort of like this pre-processing of the count data. And we're going to do some different things here. So we're going to do a default normalization where we filter some of the genes. We remove those genes that sort of like aren't expressed in any of the cells or like spots in this case. We also do this standard scaling where we subtract the mean and divide by the standard deviation. And we also do sort of like we normalize by the library sizes, but before actually doing this thing of standard scaling. And we also do some log transformations here, which I actually didn't write in the description, but that's what's happening there. We then match different scales between the observed data and the reference to make sure we're using the same sort of like distance units here, so that one distance unit actually means the same thing in different object here, being the observed data and the reference. And then we compute the landmark distances using the data and the reference. And this is a loop, so it's going to iterate across all of the different data sets in our dictionary that holds the different data objects. So we can run this. And while that's going on, I can drink some water. That's very nice. And now we're going to be looking at we're going to transfer the same genes as I showed the results of before. I think that makes a lot of sense. And those are also the genes that we included in the preprint. So it's going to say that these are the genes or features that we're interested in. And we can visualize them in their sort of like originally observed data. And for that, we again use this visualized observed, but we now specify that we want these DH genes to be actually visualized here. And there are plenty of different other settings that you can read more about if you go into the documentation. And here we sort of like see the original observed data for each of the different sections and the different genes. So that's basically what it looks like. And it looks like we've loaded data in a nice, pleasant way here. So finally, we're now ready to actually transfer the data to the reference. And we do this by this function, so like using the transfer to reference function from the fun module or methods module. And we give sort of like the undata dictionary object here as input. We specify that these are the genes that we want to use and which reference we want to transfer the information to. In this case, we're only going to run 10 epochs, but I usually use 1,000 epochs. That's more or less sufficient to get a good sort of like converged state here. And yeah, let's run this thing here. And since we're running in demo mode, it's going to be very fast. This is going to take a little while here to start up the GPU, but you see it's running through very fast. And as I said before, usually now in this case, we need to make 28 different transfers, one for each of the different features in one of the different samples or sections, I should say. And usually one such transfer, if you have a fairly decent GPU, takes about 30 seconds, give or take. So that's done here. And now we're going to do a bit of cheating, loading the pre-analyzed results here, and we can visualize the losses. As you see here, for most of the genes, it looks like we got a nice and sort of like converged state. The call2A1 gene is a bit funky here, so like that's something I'm aware of, and that's basically because it's so lowly expressed in most cases, but we can leave it as it is right now. It won't really bother us too much. It still gives fairly decent results here. And now we can visualize this sort of like transfer data here by using a function that more or less works in the same way as this visualized observed function, but now it's called visualized transfer, and that's visualized transfer data. So execute this cell here, and let's see what it shows. And here we have the different results, and we have, yeah, as you can see, all of the features, MYH6, ELN, MYH7, and call2A1 have now been transferred to each of the different sections here. And it's all living in sort of like the same spatial space. And now we can create these sort of like composite representations where we summarize the information across all of the different sections. And we want to create one sort of like composite representation for each feature, and that's always a by feature. And we run that. You get some warnings, but again, you can ignore them for now. So let us now visualize again this transfer data, but now use the composite representations. And to do that, we set attributes to composite. And there we have it. And it looks fairly smoothed. This actually looks a bit more smoothed than maybe you would have wanted it to do, but that's because we're summarizing across all the different sections. And actually, these sort of like genes that we're looking at right now, they don't really have a super strict, I guess, gene expression pattern. So that's why we get these sort of like slightly blurry images, but that's sort of like, I guess, a composite profile. So the next thing that we can do now is to sort of like assess whether we have an enrichment of certain genes in different regions here. And for that, we're actually going to use these composite representations. So we run this script here, and we use this sort of like this plot transfer here, where we look at the distribution in the different regions in the transfer data. And here we sort of like get these scatter plots where we see what the gene expression actually looks like in the different regions for each of the different genes. And you can then see sort of like this left-right asymmetry here that I was speaking of before in the call2A1 gene. And we can make a small test here where we make use of this function test region-wise enrichment from the SDA module. And we specify which region we want to compare with which other region. So it's HM1 and HM2 because that's what it says up here. And the feature we want to compare is this call2A1 gene. So we simply then run this, and we're going to run a permutation test because using any of these sort of like parametric tests is sort of like not that legit in this case because our observations are actually dependent on each other since we're using this Gaussian process to estimate the means here. So that would violate the IID assumption. So we're going to get this sort of like the result of this test region-wise enrichment, and we can just like print this message here that we test or like get an, I guess, more written statement of whether the expression is significantly different between the different regions. So that's about it for the developmental heart analysis. And I think I'm keeping time here. So we're moving on then to the mouse perturbation analysis. And this, just to remind you, is the case where we had a control case, a hem case, and a sham case. And we're just going to do something that's very similar for the first steps here, as we did in the developmental heart data analysis, loading the data and the reference. So I'm not going to be as detailed in my description of that here. So we're just going to load some paths. We then load the data, and I'm just going to sort of like rename them from having the geo terms in the data path names to having more informative names like control, hem, and sham. So that's just like what this geo name dictionary does. So we load this, and then we can have a look at it, and we see we have 3 different sections here, and obviously we would like to have maybe more replicas from each of the different cases, because right now we can't really disentangle these different sort of like case control effects from just like biological variance, for example. But again, this is just a proof of concept. So we can now just visualize the observed data, just like we did before. And in this case, we're only using 5 different landmarks. And that was sort of like, I guess, one of the questions before, how many landmarks do you have to use? And I thought that 5 landmarks seemed to be pretty sufficient for this pretty small tissue sample here. Next thing up is to load the reference image and the reference landmark coordinates, just like we did before. And we can again sort of like have a look at what the reference and the landmark coordinates looks like. And it seems like everything's working correctly. There's no mismatch between the sort of like the tissue or like the reference and the landmarks. We create this grid and the metadata that I mentioned before. So like using this reference to grid function. And this time we only have one specific region here. There's no sort of like annotation of the different regions. So I put any region to 1. And then we can have a look at the reference here and the different landmarks. And yeah, seems to be working fine. So we create this reference object using the grid that we just created. We use the reference landmark coordinates, and we use the metadata from before. And yeah, we're also gonna sort of like pre-process the count data again. You're doing this normalization and filtration, matching the scales, and also like computing the landmark distances, just like we did before. Well, what's going on? I can just mention here that so these 3 genes that we're going to be looking at are sort of like taken from one of the main figures in the publication. It's sort of like this TIMP1 gene, CD63, and terpin3N. So those are going to be our features of interest here that we specify that we want to transfer. Just like I've been double clicking something. Let me just reload the data. Let's see this. And then rerun the application. Yeah, that's great. So let us visualize these sort of like genes in the observed data. And that's the control. In the top row, we have the Hem case in the middle row and the sham case in the bottom row. And yeah, let's transfer now this data to the reference, just like we did before. We specified that it's the dictionary containing the data called MPA data. We use the mouse perturb case reference that we created, and we use now 10 epochs, but usually 1,000. And the device here just says whether we should be using a GPU or CPU device. We're going to be running this. It's 9 different transfers that we have to make here. Sorry, 10 different. We then load the pre-analyzed data, so we can again visualize the losses here, which looks like decent, nothing super strange is happening. And we can visualize the transfer data here. And here we have the different gene expression profiles for each of the different cases and each of the different genes into like the columns. And now we can actually, we're at the position where we can make this sort of like spatial differential gene expression analysis. And to do that, we're going to be using this SDA function from the SDA module. So we specify that it is the gene SERPINA3N. In this case, you can of course choose any gene that you would be interested in, that you want to analyze. We're actually going to subset, so like the reference data object here, it only includes so like those feature transfers that relates to the specific feature that we're interested in. And we want to make the comparisons across the different models. And in this case, the models is each of the different sections that we've been sort of like looking at. And this NSD is the number of standard deviations that we want to use in our differential gene expression analysis when we call a region as being non-differential expressed or differential expressed. We run that, we get this sort of like SDA res object here. And we can then do this visualize SDA result from the plotting module, where we use the reference as sort of like the first argument to our function, and then we use the SDA res object as second argument. And the other ones are just like aesthetics, I guess. And here we see sort of like the same images as we saw before, where we have, for example, in the middle part here, where there's red, there's a higher expression of the SERPINA3N gene in the Hem case, and when there's blue, there's a higher expression of the SERPINA3N in the sham case. So that's essentially the spatial differential expression analysis. And so far, we've been sort of like looking at this exact transfer approach. And now I just want to visualize or like give you a brief example of how we can use this fast approximate transfer. We don't have to transfer every single gene, but we can transfer more or less everything at once. To do that, we're going to create a new reference object, just using the same grid and the landmarks and metadata as before, but just not to sort of like overwrite the old reference object if you wanted to go back and look at that. And we're going to do something very, very similar to what we did before. We're going to use this transfer to reference function, but as you see here, it's called FA, transfer to reference. This part here, that's for the fast approximate transfer. So also for sort of like the sake of time here, I'm only going to transfer the Hem case. And here we specified the number of components, and that's sort of like the number of components we want to use in the data decomposition step. So the more the components you use, the better your reconstruction is going to be, but the longer time it's going to take to reconstruct or like to transfer all of these different components to the reference. But we run this analysis, and here it's going to take slightly more time because it's actually 25 transfers that we have to make, even though since this is considered to be one, I guess, big analysis, it's going to tell you that it's number 0 out of 1 here, so it's not going to say 0 out of 25. Let us just wait for that, but it's soon going to be done. And afterwards, we're going to run the pre-analyzed data. More seconds here. Yeah, there we're done. Load the pre-analyzed data. And now, since we transferred the whole transcriptome more or less to this reference, we can essentially visualize any single gene that we are interested in that is sort of like listed in this data set in the observed data. And here I just took the rest of those genes that were sort of like included in the figure G from the main publication, I think. So let's just define those as FA genes, and we can then sort of like visualize it. And here we sort of like have the visualization of all of these different genes, and you can put in, yeah, any gene you're interested in here. And you might wonder why aren't we like always using this fast approximate transfer? And I should just like reiterate there that we're actually seeing a loss in accuracy. It's not actually transferring the exact profiles or gene expression profiles that we see on the observed data, but it's just like an approximation of those. Just like if you were to do a data decomposition using PSA and then trying to reconstruct your data, you would get a slight loss in accuracy there. but it can be sort of like remedied by using more components, for example. And again, you're going to see sort of like diminishing returns after a certain number of components. So I think that's going to be the walkthrough of Eggplant. And maybe I can stop sharing my screen. I can actually keep on sharing my screen, but I could look if there are any questions, or maybe I can get some help with that again.
Yes, I think we have multiple questions. I can also address them again. So the one that got the most votes also on Slido was that a person missed the part of constructing the reference. So whether you could elaborate more on how to create a customized reference based on tissue type.
Yes, that's a good point. So I guess that's not really something that I included in this tutorial. I used the existing references just to save some time. And you can more or less create the reference in any way you want. So right now, it's all that you need is a image where you have sort of like your reference colored in a different color compared to the background. And I wanted to keep this reference creation process as simple as possible. I didn't really want to make that too complicated. So you can more or less use paint, just draw a figure, and that's supposed to represent your data. And I think, of course, you can also put way more effort into this if you're familiar with the tissue and know sort of like what the integral sort of like elements are and what should really be included there. That's something that you can incorporate to the creation of this reference image. But that's actually all you need, just like an image where you have a background color and something that's colored in a different way. And yeah, that's what's being loaded in here and what's then used to create this grid and then building the reference object.
I think in also a similar question. So the question was how you actually then select the artificial landmarks. So would you just pinpoint a needle on the place where you would like to pinpoint the landmark to, or how that would work in practice?
Yeah, so that's also a good question. So right now I actually built a small sort of like GUI application for myself where I load in the image and I just click on it where it's supposed to be the different landmarks. And of course, creating the reference, you should also be aware of where the landmarks are supposed to be. And I think that's sort of like that small program that I built there is something that I'm actually gonna push to the Eggplant repo to allow other people to also make a simpler annotation. But you could also use any tool. I don't know, like I think, for example, in Inkscape or in the Photoshop equivalent, you can probably see what your pixel coordinates are at different positions and just write those down in a TF3 file. So I, again, didn't want to limit this to one specific application. You just need a list that sort of like tells you which pixel coordinates the different landmarks are located at. So however you choose to do that, it should be fine. But I guess having a nice GUI for that could also be kind of like helpful. And that's something that I'm actually going to try to put in there.
Thanks, yes. The next question would be on the limits of the heterogeneity that Eggplant can handle. So in terms of tissue types, modalities, or the orientation, if you could comment on that.
Yeah, so was the last part there orientation, or what did you say?
Yeah, orientation, yes.
Orientation. Yeah, so orientation shouldn't be any issue at all. That's just like a linear sort of like, I guess, a transformation that is super easy to handle. Tissue type and sort of like different modalities. I think the modality question comes down to sort of like how well you sort of like have processed your data before. Again, we're not really sort of like super concerned about the data processing in this Eggplant function. It's just focusing on the transfer protein trying to decouple it from all of these other analysis and making it a bit modular. So maybe you could use, I don't know, harmony or SC transform to normalize or integrate your data, or this VNN approach from Sierat to then make the transfer with Eggplant. So it's supposed to be compatible sort of like with any pre-processing steps in that sense. And with respect to tissue types, I'm not really sure how I should sort of like answer that question, because I guess if we have different tissue types like liver and lung, for example, I can't really see any sort of like point in mapping those to the same reference, but I'm pretty sure that I'm misunderstanding the question here. So if I am doing that, like, please make another question, like trying to clarify what you meant there maybe.
Yes. Also a short note on that. So if you want to address your question in person, you can always raise the hand and just ask Alma directly. That also works. The next question would be on how would you determine the number of annotated regions in the reference? Right.
So yes, if I'm interpreting that correctly, it's so like how many regions you should have, or sort of like how do you define these different regions in the reference? And I think that's something that's completely up to the person who's initiating the analysis. So you can partly change introduce new, I guess, annotations to reference once you transfer the data, because the annotations aren't really related to the transfer process. It's just something that we have associated with the observations or locations within the reference, and that can be changed or re-annotated in any way we want to. And I think you should annotate your reference according to the relevant regions, or number of regions that you find relevant and what represents relevant regions to you. So that's super hard to give a general answer to, actually.
Yes. Before, you noted that you can pick the landmarks with any tool. Is there a specific tool you prefer to use for selecting the landmarks in the sample?
Yeah, I guess I should say my own small script there that I built using PyQt, but that's because I'm really bad at operating with a graphical interface otherwise. So there are probably plenty of other good alternatives there. But maybe I can push this small script up there and give a small tutorial on it. Seems like maybe that's something that people would appreciate, actually.
Yes. So the next question would be, for information transfer, is only one landmark considered, or can I use multiple landmarks together?
So yeah, again, maybe I'm not really fully understanding questions, but you include several different landmarks. For example, I think my screen is still showing here. So I have 5 different landmarks here in the control him and sham case. And all of them are used. So it's actually this function that we build where we relate the landmark distances to the gene expression. That's actually a multivariate function that includes the distance to all of the different landmarks at the same time. So to answer that question, I guess using just one landmark would give a pretty poor result. I think it's pretty easy, nice to perhaps compare this to GPS information, where we need 3 different satellites to be able to pinpoint our position. And just having one landmark would be pretty hard to use here as well. We need actually 3 different positions as well. So just using landmark wouldn't really be the best option.
Yes. What would be a good use case to use fast approximate transfer, as you mentioned?
Yeah, nice. That's a good question. So, for example, in this perturbation data set here, what we used that for was, I transferred the full transcriptome essentially to the reference, and then did this spatial differential gene expression analysis. And looking at all of the different combinations of genes, we can ask this question: what is different, for example, between the hem and the sham case? And for that, we don't really need to have an exact transfer. We just need to get an idea of which genes there is a spatial differential expression of, and then it's nice to have all of the different genes being compared and not just a select few that we need to decide on prior to the analysis. So I guess that's a pretty nice user case that we can relate to with respect to this analysis as well.
Nice, thank you. Then a general question, I think, if you would think that it would make sense to compare sections of different species, so for example, human and mouse, or different species in general?
Yeah, interesting questions, and I've gotten that a couple of other times, and I guess to some extent I'm the wrong person to ask here, because my answer would be if they seem to have a fairly similar structure, that is something that again makes sense to compare structure wise. Yeah, that would be a good analysis to conduct. But otherwise, just as it's useless to compare lungs and liver, it would be useless to compare these two different I guess species if there's no similarity or no common ground where they should be compared on. So it's totally dependent on the tissue and the biology there. And yeah, my skills aren't super good in the biology part. So.
Yes. Another question is how accurate the transfer would be between cells if the shape is really different across samples. So if you can still identify major landmarks, but the shape overall is really different. So how accurate would be the transfer be with CCF?
Yeah, so I can't really give a specific number. I don't know, not really sure how I would quantify that either. But I think, for example, this developmental heart data, and also to some extent, I guess this mouse data here shows that there can be some quite stark differences in the morphology and in the spatial structure, and we still get a good correspondence between the transfer as long as we're able to fairly accurately annotate these landmarks. So I would say, unless it's a complete distortion where your tissue is completely jumbled and ruptured, it should be able to handle quite a lot of even nonlinear distortions, and that's what we really try to design the method for, and we've done some evaluations there as well, looking at synthetic data, for example. But if people are interested, they can also look at this supplementary I get in the Eggplant bioarchive manuscript there.
Yes, thank you. Next question would be, is there a rule of thumb to prepare the tissue in order to align it with the reference later within Eggplant? So how should you ideally prepare your data?
Wow, what a wonderful question. It's someone who's actually sort of like trying to optimize their experiment for the computational analysis and not the other way around. So that's a bit of a rarity sometimes. Usually we're handed, so like we are given the data and the question is like try to solve this for us. I would say try to keep it sort of like to avoid ruptures or sort of like squeezing or unnecessary elongation. Like the more distortions that you introduce into your data, the harder it is to correct for. Like there's always something to correct for. If you can try to keep it as sort of like clean and neat as possible. But I guess that's sort of like what you always try to do. It's not someone who's like intentionally tries to fold their tissue sample. When it comes to sort of like, you know, positioning with regards to rotations or like shifts where you put this sort of like the tissue on the array, that doesn't really matter at all here. It's more about sort of like the stretching and sort of like weird distortions that I would say are more problematic. But perhaps, and one thing could be also that if you sort of like, if there is a third axis, for example, the Z axis, where you can sort of like decide where to collect your tissues from, try to take it at the same position in the Z axis, so we get sort of like a representative sort of like section from the same position. For example, the depth of the brain, we wanted to have it at the same position because it's easier to compare the structures then. But that would be sort of like my advice, I think.
Nice, thank you. The next question would be if you can use in the method actually a combination of morphology, expression, and protein landmarks, or whether you can only use one of these at a time.
Yeah, no, so that's something that we also really like that. So like, since it's completely agnostic to how the landmarks are chosen, you can choose them in using any sort of like approach that you would like to. And if you have all of these, so like modalities, protein expression, image expression, so like data and gene expression, of course, you can use all of them to guide your annotation. And I think that's something that's quite useful. I mentioned also before that we were thinking or like developing this method for automatic landmark annotation, and that's only going to be relying on the morphology and the gene expression data. But if you're doing it manually, you're free to choose so like how many or how few so like channels of information that you would like to.
Nice. Next question would be how Eggplant handles missing landmarks. So for example, if there are 1 or 2 landmarks missing in a subset of the picture, how you would handle this in the method.
Yeah, so that's a super good point as well. And it can actually handle that quite nicely. It's just gonna use sort of like fewer landmarks to actually in there, so like to actually characterize the function. So it's gonna sort of like, rather than having, for example, 5 different sort of like terms that relates to the function, is going to have 4. And if you have 1 landmark missing in one section, it doesn't really affect the other sections. What will happen, of course, is that it's going to have a slight drop in performance, perhaps, if this landmark is sort of like essential to characterize the gene expression, but it doesn't really sort of like prevent you from using the method. So it can have sort of like missing landmarks if that's the case. As long as you sort of like, I guess, when you're naming your landmarks and calling them landmark 1, 2, 3, and 4, for example, if landmark 2 is missing, don't name sort of like the other landmarks landmark 2, but you need to sort of like have a missing row there in your landmark CSV file, for example. But as long as you're clear which landmark that is which, and that is sort of like consistent across the different sections, it should be fine.
Nice. The next question, and so far as I see the last one in Slido, would be if you have 2 tissue sections, so one would be wild type and the other one a mutant, if you could still compare them even if they don't differ significantly in their gene expression.
Yeah, sure. I mean, the comparison is always possible to make. I think you could probably identify some differences, perhaps that aren't like super obvious. For example, maybe if you do a factor decomposition and want to transfer the factors instead, maybe there's some difference there. And yeah, maybe if you do this sort of like fast approximate transferring the full transcriptome, you can make something that I mentioned before. So like this unsupervised analysis, trying to identify genes that actually have a differential gene expression with sort of like respect to their spatial structure. So I think definitely that's something that's worthwhile doing.
Yes. And I think we have one more comment and one question in the chat. The one question in the chat would be how, if you have a recommendation, how the reference should be selected and how it is actually selected.
Yeah, so I think so like for me, when I'm constructing these references, I try to sort of like look at the main characteristics of the tissue, what sort of like elements or sort of like shapes seem to be present or prevalent in all of the sections that are, or in the majority of the sections that I would like to transfer information from, and then make some sort of like average representation. But you could also use like pick, maybe in the case where you actually have image data, just pick one of the images and make a reference out of that. And I didn't show that right now, But if you, for example, only working with Visium data, you don't even have to have a reference image. You can just use the spatial coordinates because that represents a grid as well. Just like we're constructing a grid from the reference image, you can use the immediate Visiums of like spots as the grid, and you transfer all of the data from the remaining Visium sections to this specific Visium section, which would then figure as the reference then, I guess. And how you choose which one of your samples you would choose to act as the reference, I guess it's up to you then. But try to make something that's as common or like as so like a present or similar to as many of the sections as possible. That would be my recommendation.
Yes. And we have one comment in the chat, and I think it would be nice to get your idea on this. So the comment was, maybe it would be a good idea or feature to visualize the displacement field between input and output to make sure no pixels move to a crazy location to match the CCF.
Yeah, that's really good. So I actually did something similar to that, if I interpret this suggestion correctly, in the manuscript, where we sort of like did this, where we can see sort of like how different, I guess, gene expression positions or patterns actually are perturbed in the sort of like the transferred versus the observed state. And I think that's hyper relevant, but maybe that's actually something that could be included here as a feature as well in the actual code base. So I'm gonna thank you for that suggestion, and think about it sort of like a nice way of implementing that. So yeah, nice. Thank you to whoever commented on that.
Yes. And I see we have one additional question in the Slido, and it was on whether you could transfer information between two modalities, in this case, between the spatial transcriptomic data and the MSI imaging data. So it would be a little different, not modalities with respect to Murphy's and Visium, but the imaging data and the spatial transcriptomic data. Yeah,
so good question. Now, I haven't really worked with this kind of imaging data, so I can't really make a super strong statement here, but I don't think it should be such a huge problem. Like you should probably be able to make the transfer. Then how you relate these two different modalities to each other, so like imaging and expression data, would be a different question that I'm not really in a position to answer right now, given sort of like a bit of a limited knowledge. But I don't think sort of like method-wise that should be sort of like an issue actually.
Yes. But nevertheless, thank you. And sure, raise the question again if you want more answers with respect to this or Alma's opinion on it. We have one additional question in Slido. So if you use spots from Visium, the question would, if you don't really lose information, if the slice is rotated or if it's measuring at different axes. So if this is not a problem for the method.
Just with the rotation, it shouldn't really be any sort of like issue here, because that's again sort of like a linear transformation. If it's along a different axis, then you would probably sort of like, it would probably be hard to, map, unless you have a completely symmetrical object, then I guess it wouldn't really matter. But if you have different symmetries around these different axes, mapping them to a 2D reference would be a bit tricky, in my opinion, and you would probably need to build this sort of like 3D reference. And there, the theory of the method is equally applicable. These distances that you measure to different landmarks can be measured in 1D, 2D, and 3D. But as of now, the software doesn't really support that. But that's something that definitely should be implemented. Yeah.
Yes. Then we have one comment. I'm not sure to which question this was raised. So maybe again raising it. So yes, maybe editing this because we have a comment. I mean, if you don't use the image as a reference, so not sure to which question this was related. Maybe you can, the person who raised it can elaborate more on this, to which question it was raised.
I mean, I will also hang out a bit in the Zulip chat, I think, if people have more questions. So yeah, I'm super happy to answer that. And if people are interested or just have questions in general about the method after this course, they can also reach out to me on like Twitter or email or anything. I think my sort of like information is sort of like included in the slides, and those will be uploaded later on. I am fairly sure of, right?
Yeah. Yes, true. We will upload everything into the Zulip, I think.
Awesome.
Nice. I think from the schedule, we still have 10 more minutes until the break. So if people want to stick around, they can ask questions. We will address them. If not, they can already head out to the break if they wish. We will continue with the schedule. I believe, let me check. So we will have from 2:30 until 3, we will have the break, half an hour break. We will be available on Zulip and here as well, and Slido to raise the questions and raise them to Alma. And after the break, we will have Vitalii joining to introduce us to cell to location. So looking forward to that. So yes, I think we raised a question again to what I just mentioned with the image and the reference. So it was in the chat, and it was, so if you have Visium, so let me phrase it. Since it was said that with Visium, we can use directly the spot coordinates instead of the reference, it was not clear to me what it meant.
Yeah, okay, so now I think I get the question. So since we're using this reference image just to create this grid using the grid to reference function, we can just as well use sort of like the spot coordinates of a given Visium sample as the grid creating the reference object later on. So instead of sort of like using the image, we can use an annotated Visium sample as our reference, or like building the reference object from that, if that sort of like clarifies the question. And there are some sort of like examples of this in the original Eggplant repository, for example, if you look through like the notebook and analysis. So the person who's interested in this, either I can explain it even more on Zulip, or they can check it out there, I would say.
Great. Yes. Yeah, that clarified it. Yes. And then we have a question where the material will be uploaded. I think we can push it to the repo or the Zulip. Both works for us well. So yeah, check out the usual communication channels and ping us if we forgot about this, but we will try to upload them tonight after the workshop.
Great. Yeah.
Awesome.
Yeah, thanks again for helping me with the questions. It's
sure.
Really nice.
Thanks for the nice talk. It was really interesting.
Thanks. I think I'm gonna mute myself and then have a look at the Zulip chat.
Yes. So if you have additional questions, Alma can also answer in the chat or afterwards on Zulip. And then I would say we can start early with the break, and we will continue with the workshop at 3 with start to location. Yes. Thanks, everyone.